Alright, so before we start, we're going to hammer down a quick difference between procs and eucs, prokaryote versus eukaryote. First little trick I like to use is pro equals no. So we are automatically going to know. The proc has no nucleus and no organelles. They're also going to be unicellular, which basically means one cell. Now they can live in groups and bundles, but they're still individual cells. If we're looking at classification, Archae and bacteria, those are the two domains. Eucs, on the other hand, have a nucleus. They've developed organelles, mitochondria, chloroplasts, all that fun stuff. They can be uni or multicellular, and they make up eukarya. Eukaryota, eukarya, kind of the same thing. This is going to be plants, animals, fungi, protists. So for now, we are going to focus on procs. We will get to eucs later, but for now, let's dive a little bit more into actual prokaryotes. Based on our tree of life, we know that there had to have been some common ancestor. Some dude way back here that branched off into all the species and created the giant tree of life. Problem is, we don't really know what this is, but we can kind of guess based on what was around. We can kind of assume that this dude right here had to live in basically trash Earth, when Earth had just huge amounts of toxic fumes and gases and the environment was very hostile. The only thing we really think this could be is some kind of Archae bacteria, because they can live in these crazy acidic or salt or cold or heat environments. We assume that the only thing that would have been able to survive and prosper was Archae bacteria. So now we can look at the two types of procs. So let's look at our two domains, Archae and bacteria. Remember, they do break down into kingdoms. The thing with this is that the kingdom is largely their name. They each have their own. Archae domain becomes Archae bacteria kingdom, and bacteria becomes the eubacteria kingdom. These are going to be our ancient extremes. Basically means they can live in the crazy places. 99.9% of the bacteria you're going to run across is going to be over here. This is what you would have on your table, on your doorknob, on your phone, on everything. So without getting too crazy on gram staining is a way we can tell bacteria apart. Inside each bacteria, or not really inside, but on the outside, there's a cell wall. So here's our little bacteria. There's our DNA floating around. Doesn't have much going on. Maybe a couple of ribosomes here and there. But that's basically what he looks like. The cell wall can have this stuff called Peptidoglycan. That's basically what the cell wall is made out of, and it's interspersed within the cell wall. Obviously, if we have a thicker cell wall and it extended out to here, we'd be able to fit more Peptidoglycan. So why do we care about this? Well, the reason is because we can use a staining tool, staining as in like staining an actual color, to figure out what's going on and use it to identify these things. So there is a stain called crystal violet. Yes, it's very fancy. You can be gram positive or you can be gram negative when exposed to this stain. This stain hooks into the peptidoglycan and basically the peptidoglycan absorbs it like a sponge. So the more of it you absorb, you're going to be kind of a blue, purple color if you can absorb a lot. However, if you can only absorb a little of that purple, you kind of get like diluted purple, which ends up looking kind of reddish pinkish. That's gram negative. So for example, the thick cell walls are going to be over here because they got lots of peptidoglycan in them. Thin cell walls are going to be right here because they can't absorb a whole lot. Bacteria can be here, bacteria can be here because they have varying thicknesses of their cell wall. Now, the big one that we really care about that can really immediately differentiate stuff is that Archae bacteria can only be over here. Because Archae bacteria does not have a cell wall made of peptidoglycan. So it will not absorb the stain and it'll just kind of be, and it'll just barely hold anything, like light pink. We don't do a lot with this, but it's easy to differentiate types of bacteria based on this because it's a very simple experiment. I can just literally drop some on there, look at them under a slide. Do they retain the color, yes or no? Gram positive, gram negative. I now have like cut down my list of bacteria that this random thing could possibly be by doing very little. So we'll go back here to our Archae and our bacteria domains, Archae bacteria, Eubacteria kingdoms. We can put always gram negative. And over here we'll put gram positive or negative. Simple, simple. Another way we can tell them apart is by what they look like. I know that seems dumb, but it's very, very simple. So we'll break this down into two categories. We will look at actual shape and we will look at organization. For bacteria, there are three shapes I really want you to look at. You can have it be a circle, you can have it be kind of an oval pill shape, capsule, doesn't really matter what you call it, and you can have it be kind of a spiral corkscrew shape. Circle is coccus, the pill is bacillus, and the corkscrew spirillum. So if you ever see a circle-shaped bacteria, that's a coccus bacteria. If it's a pill-shaped bacillus, if it's a corkscrew, it's spirillum. Now the organization, remember, since they're all unicellular, they're not creating larger and larger organisms. However, they are going to group together and kind of have colonies, essentially. So the first one we'll look at, let's say we have two coccus bacteria living in a pair. A pair is called Diplo. Say we have 4 coccus living in a strip. This is called strepto. Again, we're just kind of naming the actual organization. And the last organization is a cluster, kind of like grapes, all bound together. This is Staphylo. When I'm naming a bacteria, organization, and then I'm going to use the shape. So you may know what strep throat is. You may know what a staph infection. This is the slang term for these because they are the organization or the shape of the bacteria. If I have streptococcus bacteria in my throat giving me strep throat, that means it's streptococcus. That means it's a strip of circle bacteria. Bacteria are super tiny, so it would take way too much work to run them through super complicated tests all the time. And if we had to do that, then we would never get anything done studying bacteria. The organization lets us categorize these things a little bit easier where we don't have to run elaborate tests. We can look at them under a microscope and immediately cross out a huge number of bacteria. Then we can do more tests if needed. We can do the gram staining, we can do all kinds of stuff to further narrow it down. This is why you can get a strep throat test or a staph test relatively quickly. They can get it back to you so you know, I have this, let me take the antibiotics, let me knock this out. Because these are all bacteria. So an antibiotic would kill the biotic organisms. That'll knock this out. So it should be relatively easy to identify stuff. I can have Diplobacillus, Diplospirillum, Staphylospirillum, Streptobacillus, any organization, and then any shape. That is what my actual bacteria are going to look like. So I should be able to give you a picture of something and be like, what is this thing? And you can give me the organization and the shape. Especially if we start using crazy stuff like dichotomous keys combined with this, you can actually use the characteristics to figure out what stuff is. Not hugely complicated, but in the end, memorizing this is easy, and then we can identify and move on with our lives.